So Rahul and Peter are giving the last talk about the US NIH HubMAP consortium.
All right, hello. So yeah, Peter and I are going to talk about a project, an NIH project that we're both involved in, the Human Biomolecular Atlas program. And both of us lead components of this program, but I just wanted to mention that one of the program officers who coordinates this, Ajay Pillai, is here, and we'll actually ask him to help out a bit in questions. And him and Richard Conroy have done an amazing job of spearheading this initiative and coordinating collaboration also with the HCA. So, you know, HubMAP, the broad goals of this project, I should mention also this is a common fund initiative funded through the Office of the Director. It's a second large single cell analysis initiative that the Common Fund has had, the first being the single cell analysis program, which now moves on to HubMAP. And I think Peter will go into more detail about exactly the types of projects that have been funded, but the broad goals are very, very consistent with the goals of HCA, really to sort of accelerate and catalyze the development of the next generation of maps of the human body, to establish open data platforms, establish foundational data sets, and to really encourage collaboration across all of these different initiatives. You know, I think one of the things that HubMAP really wants to focus on, and this is of course a focus on HCA, is a vast diversity of technologies, and we've heard that in all of the different initiatives today. So certainly there'll be a large amount of single cell sequencing, but there's a significant focus on sort of spatial technologies and also metabolomics and lipid profiling and a variety of other technologies we haven't heard as much about today. And Peter will certainly go into that. As part of this focus on building reference frameworks, I talked a little bit earlier about my lab and Aviv and John's efforts to build a CCF and how that might be broadly useful to HCA, and I'll come back to that also towards the end. I'll mention a little bit about how the consortium is structured and what the overall timeline is for this initiative. There are sort of really three major complementary aspects of this program. The first are sort of the tissue mapping centers, and Peter will tell you a lot of detail about those. Those are going to be the people who are actually generating data, and there have been a number of awards funded for a variety of specific tissues and assays, and as I said, Peter will tell you about those. There's also the Hive, that's the green bar there at the bottom. The Hive is a set of 4 groups, computational groups primarily, whose job is to sort of, instead of just working on any one individual tissue, to build infrastructure and resources, for example, the Common Coordinate Framework, that will span all of the different assays and tissues that are developed. So to try to identify value and define value in the global data set rather than just looking one tissue at a time. And I'll come back to that and some of the collaborative opportunities that come out of the Hive at the end. And the last is a real focus on technology and technology development in particular. It's very likely that in 5 years, the technologies we use to think about HCA won't have even been invented as of yet now. And so HubMAP has a couple of different technology calls that are actually ongoing every year. And if you're interested in being involved, there'll be, I think, RFAs in the next couple of years as well for new technologies, both to take things that are already working and to scale them up, and also to fund new transformative technology development, again, with a particular focus on spatial interactions. You can see that a little bit here about the overall timescale. You can also see that the consortium will ramp up significantly over the next couple of years, particularly in the first couple of years now. We're sort of getting set up. We're establishing best practices and protocols. We're trying to think about how we're going to build reference frameworks like the CCF, but data generation will really start to kick into gear in the next couple of years. So hopefully that gives you a good sense of the broad structure, and Peter will tell you more about the specific projects.
Thanks, Rahul. Does the mic work? Okay, thank you. Yeah, so this is just an overview of the groups, non-computational groups that have been funded in the initial round. And essentially, we have tissue mapping centers here and technology development groups. So and here we'll have different tissues that they're studying and also different techniques they're using, and I'll try to give you an overview. So obviously, you know, sequencing, particularly, let's say RNA, single cell analysis is common across most of the groups. Chromatin accessibility as well. However, as Rahul mentioned, a lot of emphasis is being placed on spatially resolved methods, on various imaging methods, and some of this comes from mass spectrometry as well. All right, so quickly just going through the groups, there's a group funded to look at spleen and thymus. And it's basically a combination of spatially resolved transcriptomics and MERFISH and also protein analysis. And this is in collaboration with Brent Bottomiller. And so this is a spatially resolved, CyTOF technique. A group including Long Kai, Jay Shandur, and Cole Trapnell are focusing on vasculature. Again, using the spatially resolved transcriptional methods, seq-FISH in this case, in combination with the single-cell profiling of both epigenetic and transcriptional state. Richard Caprioli from Vanderbilt is focusing, and his group focusing on the kidney, and I'll show you some of this data. This is, again, imaging mass spectrometry, and gives very high-resolution data. There's a project from Mike Snyder, Gary Nolan, looking at the gut. They're using Codex, which is a multiplexed immunofluorescence, again in combination with more common molecular modalities. And there's a group from Kun Zhang, led by Kun Zhang, looking at lung and kidney. Again, combination of sort of in situ sequencing in this case, and then the molecular techniques I'll describe in a bit. The technology development groups, there's one from Long Kai again, working on improvements of seq-FISH in this case. A group from Stanford looking at lung and liver using these kind of fluorescent resonant transfer probes. Development of mass spectromic prep techniques in the context of a lung, and then also different fluorescent probes in the context of a tonsil. So I'll just give you a few slides to get you a sense for what type of a plan at least most of these groups had. Now, as Raul mentioned, the project is designed to ramp up slowly, and so most of the past year has been spent by the groups in doing all sorts of preparation, validation, both of tissue handling and the assays themselves. So here, for instance, a slide from Gary Nolan and Mike Snyder's group looking at the gut, just the sort of the sampling types, that the tissue sites that we would like to do in the initial sample, and then for year one, a fairly modest sort of production goal in terms of having one donor, multiple sites, applying the Codex, in this case Codex 2, and the single cell molecular modalities to it. This is actually a tissue mapping center that I'm involved in, looking at the lung and kidney, and the overall plan is to do the in situ transcriptomics using this DART-FISH protocol, and then also looking at both transcriptional state and the chromatin accessibility using either these assays, but there's a new assay that Kun Zhang's group has developed that looks at both modalities in the same cell. And then obviously the computational integration of this data to provide sort of a comprehensive integrative atlas going forward. Now, as I mentioned, much of the effort so far has been devoted to testing and preparation. So in this case, for instance, this means that we've generated these replicates of successive frozen sections and comparing essentially the single cell modalities that we would hope to generate in the production mode with bulk modalities that are easier to obtain and give us higher quality. And this is, I think, was a status from actually last year still. So we're moved forward since then. Now, some of the imaging modalities can be quite different, and I think we're hoping to expand and integrate this with other molecular modalities. So here's an example from Vanderbilt groups that are working on focusing mass spectrometry. So in this case, they're focusing on the kidney or using kidney as a sample, and part of it is essentially biobanked, but the other part is used for spatial transcriptomics and spatial lipid metabolomics. Now, in their case, they're using autofluorescence imaging just to provide registration and everything else, so the samples can be aligned. But then different types of assays are essentially applied. The imaging mass spectrometry probably being the most powerful and unusual one in this case, but also more traditional labeling. So here's an example of a snapshot of a human kidney. And again, the imaging mass spectrometry can span a broad range of M over Z values, so sort of 1,000 peaks resolution at about 7 to 15, I think, micrometer spatial resolution. And it can be combined in a variety of ways, both with traditional molecular imaging and autofluorescence and so forth. I'll hand it back to Raul.
So just to wrap up, I mentioned that we also have this sort of component of three different goals called the Hive, and each of the TMCs is going to focus, of course, on their individual tissue and assay of interest, and it's the job of the Hive to try to put this all together. So there are 3 general groups that I think will have many, many opportunities for collaboration with HCA. The first is the infrastructure component that's headed by Jonathan Silverstein at Carnegie Mellon. And their job is to do sort of data delivery and storage for the consortium, but also to build APIs for external developers. And we hope, of course, that they have very, very strong and close interactions with the DCP. Niels Gellenborg, who is here, and Ziv Bar-Joseph from CMU run tools components. I've copied that text incorrectly, but they'll be building tools for the processing of all of the data sets from the consortium to ensure that they're processed in a uniform way. And again, that'll be wonderful to think about what are the best and optimal processing pipelines with DCP. And then the last 2 components are headed by myself and Kari Borner, and our job is to try to build this sort of common coordinate framework for the human body, but really more generally to build reference frameworks to map to, and that includes both the spatial reference, but a set of sort of reference cell types and states that one could use as a canonical framework to apply to new technologies as those come in. I should mention that one of the things that will make collaboration with HCA, I think, quite straightforward is that many of the same people are involved in both projects. So I only have the lead PIs listed here, but for example, Sarah Teichmann and Benedict Patton are both PIs on Ziv Bar-Joseph's proposal, and of course, Aviv and John work closely with me. So there's lots of sort of natural opportunities for collaboration there, and we look forward to extending that even further. As Aviv mentioned in the beginning, there's very likely to be a joint NIH HCA meeting at some point next year, and we'll also have a joint meeting on a workshop for the CCF in the context of lung construction happening at the end of June. So thanks very much.
Thank you.